Okay. Thank you, Tomek. Thank you for the nice introduction. So, my name is Chen. So, yes, everyone. So, it's very nice to present our work for the recent past few years during my independent research in Singapore and in my current organization, which is CUHK Shenzhen. So I believe I got to know Richard Cusack in 2002 in that summer. So Richard visited Professor Douglas Turner's lab, and so we started to talk about RNA and all related research. So, and we continue to talk about RNA-related research until today. And starting in 2010, I started my own research in Singapore. So we started to work on PNA, which is standing for peptide nucleic acid. So we thought about using PNA, using modified PNA to recognize RNA secondary structures. And during which time we also continue to talk about PNA and modifications with Richard Cusack. And I got many helpful advices from him. So as we know in our human genome, so we have only about 3% of the DNA base pairs encoding for protein, and about almost 80% of the DNA sequences are encoding for RNA, and most of these RNAs, right, so are not translated into protein. So it means that we have a lot of RNAs in our cells, which are doing a lot of functions, and many of them we don't know yet. So the fact that we have so many RNAs in our human genome, in our human cells, right? So it means that we have a lot of opportunities to use RNA as biomarkers or as therapeutic drugs. So actually, people have been doing this since 1990s, right? So for example, as shown in this slide, so in 1998, the first antisense drug was developed in the clinic, right? So which is actually indeed an antiviral drug, okay? So the principle of this drug is very, very simple. So basically you can just make a 21-mer or 18-mer oligonucleotides, right, which often is modified. So this kind of sequence, short oligo sequence, can be complementary with mRNA, and so by formation of Watson-Crick base pairs with the target mRNA, you can inhibit the protein synthesis, right? So actually this antisense strategy has been recently developed, right, into a new stage, right? As you can see, starting from 1998, the first antisense drug invented in 1998, right, until now. So we have almost 10 antisense-based drugs in the clinic. Okay? So all this shows that RNA can be a therapeutic drug, but the problem is that RNA is not a simple linear sequence, right? So as shown here, we can see RNA has also folding, just like proteins, right? So in this slide, you can see from the leftmost side, you can see the sequence of a piece of RNA, and in the middle, you can see that this piece of RNA can fold into secondary structures, and based on this secondary structure, right, the sequence can further fold into tertiary three-dimensional structure. Actually, this RNA sequence is one of the first catalytic RNA that have ever been discovered in 1980s, right? So later on in 1990, I think is about 1989, right? So the discovery was awarded a Nobel Prize, okay? Sorry about it. And so we can see many more examples of these folded complex RNA structures. For example, this is a small subunit of liposomal RNA. So, and this RNA actually people have now realized that is the main target of the antibiotics for fighting against the bacterial infection. Okay? So again, this RNA is folded into very nice secondary structures. Okay? And so more recently in 2015, so in this cover of the Nature journal, so people described RNA as a weird and wonderful architecture in the cell. So the reason is that you can see RNA can fold into this very complex 2D structure, and this 2D structure in turn can fold into complex 3-dimensional structure. So, you can imagine that the folded base paired region is very hard for simple antisense strategy for targeting, right? So the goal of our research lab is to develop some ligand that can bind to this complex 3-dimensional structure. Oh, let me see. Can we go back? So you can see. Let me go back. So you can see based on the left side, you can see a green potential drug molecule that can bind to this complex secondary structure. You can see that this green small molecule, right, or maybe medium-sized molecule can bind to both the base paired region and the non-base paired region. That's our goal, right? Eventually, I will show you how we can do this. Okay. So first of all, right, binding to the unbase paired region is simple, right? You can just use the antisense strategy. So how can we do the targeting of already base paired RNA regions? So we can learn from nature. Actually, in nature, right, we have already seen many complex structures, including the triple helical structure as shown here, right? You can look at it as a rotating molecule. So the black and the red strand can form base paired duplex, right? And then you can allow the yellow third strand to bind to the major groove of this duplex structure. So in this way, you can simply design one piece of molecule, right? So for example, you can use another RNA molecule dependent. So that in this way, it allows us to recognize the duplex structure and the sequence. So when you look at the triple helical structure, you can see there are 3 bases in each layer, right? So if you look at each layer, you can see a U base pair can be recognized by another U base on the left, top left side, right? And in the middle, you can see by simply changing the oxygen at 2 position to sulfur, yellow sulfur, you can enhance the binding. So this sulfur is larger and is more polarizable. So you can allow more stable formation of this base triple interaction. And the problem with this base triple recognition principle is that when you have a GC pair, so you cannot use normal C, like unmodified C, because C has N3 unprotonated usually most of the time, right? So that means you have to lower the pH to protonate the N3 nitrogen of this C to allow the formation of 2 hydrogen bonds between protonated C plus and the G in the middle strand of the RNA. So that's one of the problem with targeting RNA by triple helical structure formation. So later on, I will also tell you that some other problem. For example, as you can see in this triple helical structure, all 3 strands are negatively charged with phosphate backbone. So that means you have intrinsic instability for this triple helical structure. Of course, in nature, this triple helical structure still can form. That is because this triple helical structure is within a very complex tertiary structure scaffold. For example, a pseudoknot structure. Right? You can see we have these 3 examples of these triple helical structure in nature. So all of them can form through the pseudoknot structure formation, sort of. Right? For example, the telomerase RNA, the SAM2 ribose switch, and the other one is the pan RNA that can be found in actually also in viral RNAs and also cellular RNA. So this pan RNA is basically formation between poly A tail and the U-rich internal loop. You can see that these U-rich internal loop can accommodate the poly A tail sequence, then allows the formation of the triple helical structure. Of course, this triple helical structure is stabilized by both Watson-Crick UA and Hookstein UA base pairs. Right? So, but for us, if we want to recognize preformed base pair RNA duplex structure, so the triple helical structure will be only stabilized by our third strand and interaction with the duplex preformed structure. So in the beginning, we started to collaborate with Kiyosaki lab. Right? So for example, he provided us this S2U modified triple helix forming oligonucleotide. Right? So for example, in this case, right, on the right side, you can see one hairpin structure. We named it as RHP1, and RHP1 can be recognized by this red strand. So with a sequence of UC, 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 and we can modify some of the U, right, into 2-cell U by simply changing the oxygen to sulfur. And we show, as you can see in the screen, in the top left figure, right, you can see without any change of the TFO sequence, right, the TM is quite low, which is below the human body temperature. Right, the TM is 28.1 degrees Celsius. By simply changing one atom of this TFO strand, right, so you can see the second one, right, the TM is increased from 28 to 40 degrees Celsius. And by further replacing more U with the 2-cell U, you can see the melting temperature increases further. And so you can see this is quite promising, right? By simply modifying the base of the third strand, you can enhance the binding between the third strand and the preformed duplex structure. But the problem is, again, you can see, as I already mentioned, so this thermal melting study was done at pH 6.0, which is not really a physiological pH. So that means we have to solve this pH problem. So another problem is that when we tested the same TFO sequences, these 4 sequences, for binding with the same DNA sequence, right? We can see that the TM is much, much higher. You can see in the table again, right? You can see 2 columns. Right? One is for the TM with the double-stranded RNA. The other one is for the TM values when you bind the TFO with the double-stranded DNA, right? So that, but the goal of our research group is to target the RNA in the cell. So if your TFO binds to the DNA more strongly than RNA, that's a problem, right? So we have to overcome this problem. So then we started to look around, right? So because this sugar phosphate backbone doesn't work, right? We also tested locked nucleic acid provided by Kiyosaki lab, and the RNA also shows the same problem. So then we also start to think about this, right? So of course, DNA duplex and RNA duplex are different, although the base sequences and the base structures are almost the same, right? Okay, but the geometry of A-form RNA duplex and B-form DNA duplex are very, very different, as you can see here, right? So RNA duplex is much shorter and thicker compared to B-form duplex. And another parameter you can easily see is that the rotation, right, of the A form is much smaller compared to B form. You can see each base pair rotates about 36 degrees for B form DNA, but it's only about 33 degrees for A form. So this twist and the rise, right, the difference between B form and A form may allow us to find some molecules that can selectively recognize A form duplex instead of B form duplex. And so, yeah, so then we started to use PNA as shown on this screen. So as I just mentioned, so before this, we also tested 2' of methyl RNA, right, and modified RNA with both base and sugar modifications, right? For example, the LNA. So all of the LNA 2' of methyl RNAs bind to double-stranded DNA more strongly than to the double-stranded RNA. So with them, we started to use this so-called PNA molecule. So PNA stands for peptide nucleic acid. So actually, this is not an acid molecule. As you can see, there's no acid group in this molecule. So just want to mention here. So here the letter B stands for the different bases, nuclear bases that are attached to the different backbones. So when you look at this PNA molecule, you can see that for each unit, you have the same number of atoms as the normal DNA or RNA. For example, you can see here. So for 1 unit of DNA, you have 1, 2, 3, 4, 5, 6. You have 6 atoms in 1 nucleotide. And in PNA, you have also the same number, 1, 2, 3, 4, 5, 6 atoms. Although in PNA, you don't have a cyclic sugar structure, and all the atoms are very, very different. But based on the design, right, which is already 30 years ago in 1991 from Peter Nielsen's lab. So they originally believed that this PNA molecule can bind to double-stranded DNA. And later on, we and others showed that actually the PNA molecule can bind to double-stranded RNA molecules more strongly compared to double-stranded DNA molecules. So the reason may be probably because, right, so PNA is less twisted, and which allows it to selectively bind to double-stranded RNA more strongly compared to double-stranded DNA. Okay. So of course, PNA also have many other advantages compared to normal sugar phosphate-based oligonucleotides. For example, it doesn't have any sugar phosphates, right? So it is neutral, as you can see in the structure here. Another is that this molecule is not a natural unmodified nucleic acid. It's not a natural peptide. So this molecule is chemically stable. It's not recognized by either proteases or nucleases. So it's chemically stable. And so there have been many animal studies showing that PNA is not toxic, and it doesn't show immune response. So so this is probably because it's a neutral molecule. It's not recognized by protein molecules, so there's no immune response. So, and yeah, actually, right. So I just want to show you more recent animal studies, right? So as you can see in these 3 papers, right, people have showed that these molecules are very promising in the animal model studies with anti-cancer activities. And these PNA molecules are mostly modified in the backbone to improve the solubility and also improve the cellular uptake. For example, in the second one, you can see is so-called mini-PEG gamma-modified PNA. So here it means that people can attach the short PEG moiety into the backbone, right, to pre-organize the molecules, and it can show more enhanced binding. Yeah. So in these 3 examples, right, the first 2 are for targeting microRNAs, and the third one is for targeting mRNAs. Right? As I just mentioned, so many RNAs have their own secondary structures. Right? Although we can see these binding property and anti-cancer cellular activity and animal models activity, but the problem is that if the molecule, if the target molecule has its own secondary structure, so it definitely will weaken the binding, right, between PNA and the target RNA molecule. So that comes to our goal. Yeah, so we want to modify the PNA so it can recognize the RNA structure. And another example is that actually recently, maybe about less than 5 years ago, people developed this company called NewBase, and this company actually is now in the preclinical stage, and I heard that they are trying to put the PNA molecule into clinical test probably sometime next year. Okay? So yeah, so we are seeing that after almost like 30 years of development, PNA probably will also come to stage to be serving as a promising therapeutic drug molecule. Yeah. So we are very excited about this, and at the same time, we are also doing something different from what Peter Nielsen and Dennis Lee are doing. So basically, the goal of our research lab is to develop this modified PNA shown in red color here that can selectively bind to the already structured RNA duplex. Yeah. So, but not binding to the single-stranded sequence of the RNA, which are often present, which are always present in the RNA secondary structures. So this is our goal. So we started publishing some papers in NAR and other journals in 2004. It took us some time. Yeah, although our lab was already doing this PNA-related research in 2010, but it took us some time to finish a story and to publish a paper. Yeah. Okay. So yeah. So yeah, for the past almost like 10, 11 years, we have been doing this. So basically, yeah, it can be summarized in this slide. Right? Basically, we have a piece of the RNAs, and which can have a duplex region. And if we look, consider the orientation of the base pairs, we will have 4 base pairs that are AU base pair and UA base pair, and the GC pair and the CG pair. So if we want to recognize an RNA duplex sequence, we have to recognize all these 4 base pairs. So far we can summarize that. We can use these 4 different bases to recognize AU, UA, and GC-CG base pairs. So basically, we can use 2-cell U to recognize AU, and here we have this letter R. So this is for standing for this special R monomer that can recognize the GC pair. And another shown in blue color here is the S monomer, which is designed based on the published work on this DNA targeting research. Yeah. And another one is Q base that can recognize the CG base pair. So I will further discuss these different base pairs design and thermodynamic characterization. Okay. So yeah, more recently we also developed many more base pairs. So you can see we have modified the U base with halogenated bases. Like for example, this is bromo U shown in green color, and 2-cell U also shown in green color. So in the number shown in the parentheses are the year in which we published the work. Right? So for the past few years, so we have published a few papers reporting our efforts in recognizing the base pairs in double-stranded RNA. Okay, yeah. So we also did some modeling study. So as you can see here, the PNA strand is shown in this yellow color. So this atomer PNA can bind to the preformed RNA duplex, which is shown in this cyan and green color. So you can see the yellow PNA strand binds into the major groove and recognize the sequence of the RNA duplex. So we name this as a DBPNA, so which stands for, maybe I can go back, right, which stands for double-strand RNA binding PNA. So this is why we name it as a DBPNA, double-strand RNA binding PNA. Okay, right, so let's continue. So, so first of all, I want to tell you about how we started to think about designing this so-called L monomer, L base, right? So actually L base is not completely new, right? As you can see, compared to previously designed J base, right? So the difference is only one atom. So we simply changed the oxygen in this J base into sulfur in this L base, right? So again, just like a two-cell U base, right? So by changing the oxygen to sulfur, we can enhance the van der Waals contact between this L base and the J base, the G base in the GC pair. Another advantage of this sulfur substitution is that you can selectively recognize G in this Hooke's-Steen edge instead of the Watson-Crick edge of the G, right? Shown in the bottom right corner, right? So we have, if we want to form this Watson-Crick-like G-L pair, we will have this very significant static clash between amine of G and the sulfur of this L base. So, and if you put this L bases into a piece of PNA, like this blue PNA strand, you have this selective binding between PNA strand and the duplex RNA, but no binding between PNA strand and single-stranded RNA because you have this static clash. This is a geometric consideration, right? So it sounds very nice. And compared to J, it's advantageous because J base is already shown to be able to bind to GC pair and also able to form JG Watson-Crick-like pair. Okay, so we have to prove that by experimental data. Actually, we did that. So first of all, we did some thermal melting experiment again. So we made these three PNAs, right? So the first one is L modified PNA, TL, TL, TTTL, and the middle one is J modified, and the last one is unmodified PNA. So we want to compare the properties of C, J, and L within these three PNA strands. Okay. So we can see that the binding of the first L modified PNA actually is that you can see the L modified PNA shows the highest TM, it's 64 degrees Celsius, and unmodified PNA shows the 38 degree, and the J modified actually shows the weakest binding. And when we tested the binding to double-strand DNA, so we see no binding at all. Right? Okay, that's very good, right? So, and when we test the binding to single-stranded RNA, and we can see the opposite trend. So unmodified PNA shows the highest TM, and our L-modified PNA shows almost no binding. Right? Okay, and when we test the binding to single-strand DNA, also we see the similar trend. So you can see based on these PM values, we can see that our L-modified PNA can show selective binding to double-stranded RNA over double-stranded DNA and single-stranded RNA. And also similarly, we also tested the binding between Q-modified PNA and RNA structure. We will see that the Q-modified PNA has selective binding to double-stranded RNA over single-stranded RNA because there's a steady clash between QG pair if they form the Watson-Crick-like pair. I have to speed up. And also show that the binding is happening at pH 7.5 instead of pH 6.0. Okay, yeah, which is much better than traditional TFO. We also tested binding to many other RNA and DNA structure, and we see that the binding is only happening between the target RNA duplex and our PNA. And there's no binding between PNA and other RNA and DNA structures. So only one binding event between the target molecule and our PNA. Okay, and also if we have a multiple Q-modified PNA, we can see that the PNA can get into the cell easily. You can see that in this confocal data. And I just mentioned that you can change T into 2-cell U to enhance binding, right? So, and we also tested this, what can happen if we do this in the PNA sequences. And as you can see that comparing the sequence on the left and the sequence on the right, the binding can be enhanced by tenfold. Like the KD value increases from 4.7 to 0.4 micromolar. Right? So by changing T to 2-cell U, we can see that we can see significant enhancement of binding. So that gives us insight that in the future we can simply do this to enhance binding. So then next I will talk about some applications of our currently modified dsRNA binding PNA, in short dbPNA. So the first question we want to ask is whether we can design our PNA, dbPNA, that can bind to duplex region of this viral RNA in influenza virus. So in this project, we collaborated with L. Cusack, as you can see the reference. So because in this RNA, we have this very conserved region, so-called panhandle region, formed between 5' end and 3' end of this viral RNA. And this is almost 100% conserved in influenza A. And if we look at the sequence, you can see that this is the shortest segment of this viral RNA, and it can form this so-called panhandle structure, and the 5' and 3' form a duplex. It's about 25 nucleotide region. Okay. If we zoom in this region, the duplex is formed, stabilized by these multiple base pairs, and we select this region, right, for the design of our modified dbPNA. So basically, in this case, we designed this TAM PNA, right? So TL, TTT, QT, LLL, right? We use this Q to recognize CG, T to recognize AC and AU pairs, and L for recognize GU pairs. So instead, it works, right? So you can see the binding of 300 nanomolar in this physiological pH and salt condition. And we also tested whether the
We have some issues on the connections. Can you hear us?
Yeah, it's okay now? Can you hear me?
Yeah, we lost you for a moment.
Okay, sorry about this. There's a connection problem here. I cannot see my slide. Okay. Where's my slide? Okay, thanks. Yeah, okay. We got to low, please. Yeah. So to do that, we modify the PNA, by this way, right? So we simply added this benzosulfine into this T base of the PNA. So this modification allows the PNA to be fluorescent, right? So we named it BTU. Yeah. So in this case, we titrated the both the single-stranded RNA and the double-stranded RNA into the PNA, and we imagine that because this PNA now is fluorescent, we can see some fluorescent properties. Indeed, we see that. Right? From on the left side, we can see that titration of a single-stranded RNA into the PNA shows no response. The PNA fluorescence stays at the same value. Right? And on the other side, you can see the titration of this double-stranded RNA, we named at PHV, panhandle viral RNA, into this PNA. You can see enhancement of fluorescence. So based on this fluorescence data, we can fit the curve and get a KD of 700 nanomolar, which is similar to the gel shift data. So So based on this data, like we can see that our PNA is selectively binding to double-stranded RNA over single-stranded RNA. And we imagine, so why the PNA shows the enhancement of fluorescence upon binding to RNA to form triplets. So probably because based on the modeling data, we believe that it's because in the triplets, this benzyl sulfine group is more exposed and the fluorescent property is changed compared to when it's in the single-stranded PNA structure. Another example is that if we compare this the L base, right, so and the J base, right, you always have this guanidine moiety in this structure, right? So that's why it inspired us to develop this so-called R monomer. So we simply connect the guanidine group with a 2-carbon linker, and then we make this very normal PNA monomer, right? And based on this binding data, we can see that this R-modified PNA can actually also bind to the viral RNA structure very tightly with a KD of 200 nanomolar. And based on the geometry, we also can see that the R-modified PNA doesn't want to bind to the single-stranded RNA because R and G shows a steric clash and with no more than 2 hydrogen bonds. Right? Actually, there's only 1 hydrogen bond can form between R and G. If we want to form RG Watson-Crick pair. And we also use some fluorescence binding assay to demonstrate this. So here, right, so on the left side, you can see this R-modified PNA. That is a model PNA, right? And we also use this so-called 2-aminopurine modified PNA. So using a number 2 representing the modified RNA. So 2-aminopurine is fluorescent, right? So when we titrate the PNA into the RNA, we can see fluorescence change if there's binding. Indeed, we see the quenching of the fluorescence, probably because there's a stacking between this T and this 2-aminopurine. So you can see the binding can occur between this target RNA molecule, this RNA duplex, and the PNA. The KD is about 0.4 micromolar. You can see the black curve. And all the other points are for the binding between the RNA when the GC pair is changed to CG, AU, or UA. So you can see there's no binding. And when we test the binding between single-stranded RNA and these R-modified PNA, we see very weakened.
Again, some issues with the connection. Welcome back. Sorry, we have more issues. Hello. Professor Chen, can you hear us?
So can we continue?
Yeah. Sorry.
All right, sorry about this.
So yeah, we can hear you. You can go on.
And another point I want to mention is that actually this so-called panhandle viral RNA duplex structure is not accessible by traditional antisense molecule, for example, DNA molecule, and there's no binding if we want to titrate the DNA into this structure. Yeah. So okay. So to deliver the PNA into the virus-infected cells, so we decide to conjugate the PNA with a sugar molecule called neomine. Yeah. And indeed it works. So when we conjugate the neomine and attach the PNA molecule with fluorescein, so we can see that the green color labeled PNA is inside the cell. Right? When it's inside the cell, it's also homogeneously distributed inside the nucleus, and also is also inside the cytoplasm. So that's very important because this influenza virus replication is happening inside the nucleus. Okay, so this is very, very encouraging. So we also tested the antiviral activity, and indeed, I just go to the next slide, and we can see that when we apply 5 micromolar neomine attached PNA, our dbPNA into the influenza virus infected cells, we can slow down the virus replication by at least 10-fold. Okay. Yeah. So again, we come back to this RNA secondary structure cartoon, right? So because for the previous part, right, I just mentioned how we can design our dsRNA binding PNA for targeting double-stranded RNA, right? So how about the nearby single-stranded region, right? As you can see, secondary structure of RNA is always composed of secondary this single-stranded region, double-stranded region, single-stranded region, double-stranded region with repeating different motifs. So as again, we can combine, right? It's a simple idea is just that we can just combine the dbPNA with traditional antisense PNA, right? So, and we did some modeling for designing this combined PNA scaffold, so-called DAPNA, right? So we combine dbPNA with antisense PNA, right? So the dbPNA shown here is a blue color, and the traditional antisense PNA shown here is a green color. So we just simply connect antisense PNA with dbPNA. We can recognize simultaneously the double-stranded region and the single-stranded region. And the modeling shows that this can be done. Of course, this is done in the computers, right? So we have to do it in wet lab. Okay, let me see. Yeah. So and also we also did some other combinations. For example, DAPNA targeting different double-strand, single-strand RNA regions, right? So and our modeling shows that DAPNA can only bind to the first 2 different RNA secondary structures. That means that the DAPNA can form parallel triple helical structure and anti-parallel duplex structure. So it's orientation and sequence dependent. That it means that this DAPNA scaffold is very, very promising in recognizing complex secondary structure within a large RNA, including virus RNA. Yeah. Okay. So we first tested some simple hairpin again. You can see here there's one PNA strand with a blue color for dbPNA region and with green color for antisense region. Right? And this DAPNA can recognize this hairpin that is found in tau mRNA region. Right? So we decided to use this blue dbPNA for recognizing the top stable base paired region and the green antisense PNA region to recognize the weakly formed bottom part of this stem loop structure. Right, so you can see based on this complex RNA PNA structure, you can see we have parallel triplets and anti-parallel duplex forming. Indeed, we can see based on this gel shift data, we can have very tight binding up to 40 nanomolar, right? And we also tested some control PNA. For example, we have this blue region only, and this is for DBPA alone, right? And also DAPA with the Q base replaced with another E base, so we see no binding. And we also finally we test this all green color antisense, traditional antisense PNA in the bottom of the gel shifted data. We can see the KD is comparable to our DAPNA, right? Slightly tighter. So, but the advantage of our DAPNA is that it can recognize both the sequence and the secondary structure of the target RNA. But the traditional antisense PNA, although it's binding is tight, but potentially it can recognize many other sequences, right? Or with different structure. Right? So, but our DAPNA is that is relatively selective. It can recognize this specific stem loop structure present in the pre-mRNA. Okay, so I will finish here and with a summary. So basically, I hope I have convinced you that we can use our so-called dsRNA binding PNA, dbPNA, for targeting secondary structures of RNA with structure selectivity and sequence specificity. And our dbPNAs, if conjugated with a neoamine molecule, can be bioactive, right? It can modulate functions of or inhibit the functions of virus replication. And in the end, right, so if we combine these 4 letters of dbPNA, right, with another 4 letters of antisense PNA, we can get a total of 7 letters for this so-called DAPNA. So although I didn't show a lot of data for the DAPNA scaffold, so eventually we believe that we can use this DAPNA, this 7-letter platform that can be used to target many virus and cellular RNA molecules. Okay, in the future we hope to collaborate with you guys and also QZ lab, right, to demonstrate the activity in animal models and to demonstrate the real target is the one we want to target, right? Okay. Finally, I want to thank my current group in CUHK Shenzhen and also my former group members, right, for the past few years. So we have been doing this, right? And also, of course, special thanks are given to Professor Richard Cusack. His name is shown in the collaborators list. Okay. Finally, I want to thank the funding agencies and our universities for supporting our research. Okay, I'll end here. Thank you.
Thank you, Professor. Now it's time for questions.
So, can you hear me? Yeah.
Yes,
thank you for the presentation. I have a question regarding what you mentioned about toxicity of PNS, because you said they are not very toxic or little toxic. So I'm wondering in the perspective, so can you make them toxic? And because if you want to apply them as a cancer drugs, you actually want to kill cells. So if you can comment on that.
Oh, yeah. So of course, we have done some cellular anti-cancer studies. We can see some anti-cancer activity. For example, we have made a PNA that can bind to microRNA 21 precursor happy structure. So there's some anti-cancer activity we can see. When I talk about toxicity, I mean it's there's no off-target binding causing toxicities. So of course, if we want to make our PNA designed for targeting very important RNA molecules, which is critical for cancer or even for virus, right? So we can show this activity. Yeah, we can see this activity. Yeah.
Yeah. So you can make kind of structure-related activity for making them.
Yeah. Yeah. As I shown in this, I only showed the antiviral activity in this talk, right? So yeah. So we it's important to mention that this special PNA, right, is not toxic in the cell for the cell, but it's toxic for the virus, right? So we can inhibit the maybe LQZ can can give more comments, like, because she and her colleagues did the antiviral study. So based on her data, right? So we don't see very significant cellular toxicity, but we see this tenfold decrease in slowing down the virus replication. Yeah. So we can, of course, So depending on the target, right? So we can make the our PNA, right, that can be used to regulate the functions of viral or cellular RNAs.
Okay, thank you very much.
Thank you. Is there any more questions? I don't see any questions from the online viewers, so thank you once again, Professor Chen.
Okay. Thank you. Thank you, guys.